So we're going to start doing Chapter 17. Everything from here on out will be on the final exam. So Chapter 17 is about the cytoskeleton and motor proteins. Very interesting stuff. You guys already know about my love for the ATP synthase, the complexity of that molecular machine. But these motor proteins are just as interesting, really complex molecular motors, machines at the molecule scale. So they're really pretty impressive proteins. And then the cytoskeleton, the superhighways that they walk down, whether it's the microtubules for kinesins and dyneins or actin for the myosin motors, also very interesting. There's a lot of dynamics going on, making them larger and smaller. So the first thing that we want to talk about is the major components of the cytoskeleton. So what are the microtubules, what are the intermediate filaments, and what are the microfilaments? And then we're going to talk a little bit about how intracellular movement takes place, how the muscles contract, or how the vesicles are delivered inside of the endomembrane system, the motor proteins moving inside of the cell. And so those are going to be our goals for Chapter 17, so let's get started. This whole need for understanding this was discovered pretty early, and so it's pretty obvious that if the cell body is where the ER and the Golgi are, then proteins get made there, but the neuron can be feet long. If we think about the sciatic nerve in me is 3 feet long, and in something bigger like a whale, a giraffe, elephant, the need for moving things from the cell body up in the horn of the spinal column, all the way down to the axon terminus at the neuromuscular junction. The need for moving these proteins this distance is going to require some sort of process that diffusion and Brownian motion is not going to be able to handle it. So there must have been some molecular motor doing this, and so experiments were done to radio label proteins and look at where they were over time to show that there was a processivity, some sort of movement of these vesicles from the cell body out towards the axon terminus. And so we wanted to find out, what those motors were and how they were moving. Around the same time, the advent of electron microscopes started elucidating the cytoskeleton, and so the cytoskeleton comes in pretty much 3 flavors. There's the microtubules, which are the biggest, 25 nanometers in diameter. They're really big. And then the microfilaments, which were pretty obvious from even light micrographs of striated muscle, that they were the thin and thick filaments. And then intermediate filaments are everything in between. And so we started investigating the microtubules, and what we found was that they were a complex of proteins. So there's the tubulin dimers, the alpha and beta tubulin, create a heterodimer, and that is the subunit that makes up the protofilaments. So it'll be a dimer followed by a dimer followed by a dimer will make up a protofilament. And then 13 protofilaments will come together to create a tube that we call the microtubules. And so you can see through these EM micrographs here in this picture that we can see these this structure, and so we're pretty sure about this structure of microtubules. And microtubules make up or are important for several things. The microtubules seem to, in animals, originate at the centrosome, and so animals and fungi have a centrosome that microtubules are growing out of. Inside of the centrosome are these things called centrioles, and the centrioles are in many eukaryotic cells are the microtubule organizing center. This isn't the case in plants. Plants don't have centrioles. They just have a microtubule organizing center that performs pretty much the same function as the centrosome. So the centrioles, there's always two of them in the cell. They get duplicated in S phase. Same time you're duplicating your DNA, you duplicate your centrioles, and they are always like in this perpendicular arrangement to each other, and they are made up of microtubules, and they are made up of 9 sets of 3 microtubules. So 9 sets of 3 microtubules make up a centriole, and you have 2 centrioles at the center of your centrosome, and the centrosome is just a jelly-like substance around the centrioles. So this 9 times 3 arrangement of microtubules is true of centrioles, and it will also be true of another structure I'll show you in a second. But that's something to remember. It's often on things like MCAT tests or NCLEX or some of these GREs. What is the arrangement of microtubules in the centrioles? And it's a 9 times 3 plus 0 arrangement of microtubules. And like I said, they exist inside of the centrosome, which is just a jelly-like substance around the centrioles, and that's the organizing center for the microtubules coming out of the centrosome. So the cytoskeleton, at least the microtubule portion of the cytoskeleton, originates at the centrosome and moves out from there. And microtubules are important in several cellular functions, like they give the cell structure. They are the superhighway system that motor proteins are walking down to deliver vesicles from one part of the endomembrane system to another. But they're also really important in dividing cells. They are what creates the spindle that gives the motive force for the separation of chromosomes at anaphase, and actually aligns the chromosomes at metaphase. And so they're going to be really important in the dividing cells. And then they're also important in the movement of cilia and flagella. And so ciliated cells, like the ones that line your lungs and create the mucus elevator, are the ciliated cells are using microtubules to create that ciliated motion. That's how paramecium get around, for instance, through cilia. microtubules have something that we call dynamic instability. And so what that means is that microtubules are always growing on the plus end. And the way this works is when a tubulin heterodimer is GTP bound, then it can add to the positive end. So the positive end is constantly growing by the addition of GTP bound tubulin dimers. And so this one side of the microtubule is growing, and so we call that end the plus end because you're always adding to it. This GTP gets hydrolyzed. The GTP hydrolysis just happens on a sort of a clock, and so the GTP hydrolysis is progressing at the same rate that the microtubule is growing, and because of that addition of those dimers, there's always a GTP bound tubulin dimer cap. There's always this end has GTP attached to the microtubule dimers, tubulin dimers. However, if GTP were low for some reason, maybe some kind of signaling process, or maybe just energy, for whatever reason GTP gets low, then that hydrolysis of the GTP might catch up, that there were no GTP bound tubulin dimers to add, and the just constant hydrolysis would make it to the end. Once there's no GTP bound tubulin cap, then the end is no longer stable. The positive end is no longer stable, and it starts releasing tubulin from the end. And this is a very rapid process called catastrophe. If the GDP front ever catches up with the end, then catastrophe happens, and then you have your microtubules shrinking. So they're constantly growing as long as GTP is around, but when there's no GTP around, then you have this shrinking called catastrophe. On the other end, the non-growing end, we call that the minus end. And at the minus end, there's another different kind of tubulin called gamma tubulin. And gamma tubulin is all studded into the centrosome matrix. And so it forms this ring complex from which the microtubules can grow. And so that's the dynamics of microtubules. They are constantly growing and shrinking in a GTP dependent fashion, and that creates this polarity of plus end and minus end. And then the motor proteins are going to walk down the tubulin.